Okay, so you've got that big chemistry test coming up, right? Chapters 9 and 10. Well, we're going to take a deep dive into all that good stuff from your study guides and textbook excerpts. Pull out the need-to-know info.
Yeah, and we might even uncover some really interesting stuff you can, use to impress your teacher.
All right, so first off, let's talk about carbon. This element is like everywhere.
Oh, absolutely. I mean, it's in over 90% of known compounds, which is pretty mind-blowing when you think about it. It's like the backbone of, well, almost
Yeah,
everything.
like the clothes we wear, the food we eat. Crazy.
Exactly. And the reason carbon is so special is because of its bonding ability. It can form four bonds. So imagine it's got like four arms, each reaching out to connect with other atoms.
So it's kind of like the ultimate Lego brick, right?
Exactly. It can create these incredibly diverse and complex structures. And get this, those bonds between carbon atoms, they can be single, double, or even triple.
Wow, so that's a lot of variety.
Oh, yeah. And you can actually see this if you look at ethene, ethene, and ethane in your textbook. Ethane has a single bond between its carbon atoms. Ethene has a double bond, and ethane and triple.
Okay, so like real-world examples of carbon in action.
Well, think about hydrocarbons, those compounds made of carbon and hydrogen.
Right.
Those are the basis of fuels like gasoline
Mm.
and natural gas, the plastics that are kind of everywhere these
Oh,
days.
yeah, and even some clothes are made from them too, right? Like polyester?
Precisely. And you know what's really interesting? Those clothes might have a connection to, get this, ancient ferns and dinosaurs.
Wait, what? How?
Well, fossil fuels, which are the source of many hydrocarbons, they formed over millions of years from the remains of organisms that lived, a really long time
Well,
ago.
that's wild. So basically, we're wearing dinosaur clothes.
In a way, yes, it all goes back to carbon.
So carbon is amazing, but life obviously needs more than just carbon and hydrogen.
Right. That's where biomolecules come in. Those are things like carbohydrates, proteins, lipids, and nucleic acids.
Okay, and these are much more complex, right?
Oh, yeah. Think of them like intricate structures built from smaller building blocks called monomers. Like, imagine a chain made of individual links.
Okay, I'm with you.
So carbohydrates, for example, those are made up of sugar units, like starch.
Ah, starch, like in bread and potatoes.
Exactly. And when you eat those starchy foods, your body breaks down that starch into glucose, which your cells use for energy.
Okay, that makes sense. But then why can't we digest cellulose? I mean, that's made of glucose too, right?
That's a great question. And the answer is all in the way those glucose units are linked together. Starch has the specific type of bond that our enzymes can break down, but cellulose, nope, different bond. Our enzymes can't handle it. It's like trying to fit a square peg in a round hole.
Ah, okay. So cellulose passes through us as fiber.
Right.
Okay, so let's talk about proteins. They're like the workhorses of our cells, doing all sorts of things.
Absolutely. They speed up chemical reactions, transport molecules, fight off infections, you name it.
So they're like little tiny machines.
Yeah, that's a great way to think about it. And what's amazing is that their function is so closely tied to their shape. Each protein has a very specific shape that allows it to do its job.
Okay, and that shape is determined
By the sequence of amino acids, those are the building blocks of proteins. So the order in which those amino acids are linked together, well, that dictates how the protein folds and twists, creates that unique 3D structure.
uh,
It's like origami almost. And you can actually see this with hemoglobin, which is the protein that carries oxygen in our blood.
all right.
It's got this perfect shape for binding and transporting oxygen molecules. It's pretty incredible.
Okay, moving on. Lipids, which include fats and oils.
Right. Lipids are super important. They're like the energy storage units of our cells. They also play important structural roles, like forming cell membranes.
Okay, and there are different types of lipids, right?
Yeah.
Saturated and unsaturated fatty acids.
Yeah, you got it. And you can tell the difference just by looking at them. Saturated fats, like butter, they're solid at room temperature. But unsaturated fats, like olive oil, they're liquid.
And why is that again?
It all comes down to their molecular structure. Saturated fatty acids have straight chains, which lets them pack together really tightly.
Yeah.
But unsaturated fatty acids, they have these kinks in their chains, so they can't pack as closely.
Oh, okay. And what about phospholipids? I remember hearing about those.
Yes. Phospholipids are super important for cell membranes. They have this really unique structure. It's got a water-loving head and a water-fearing tail, which allows them to form a double layer that acts as a barrier, controlling what goes in and out of the cell.
Oh, that's cool.
Yeah, they're like the bouncers of the cell, deciding who gets in and who stays out.
Now for the big one. DNA.
Ah, yes, the molecule of life. It's got that iconic double helix structure, and it's basically a library of genetic information containing the instructions for, well, for building and maintaining an organism.
And it's made up of building blocks called nucleotides,
Yeah.
right?
Yes, exactly. And each nucleotide contains a sugar, a phosphate, and one of four nitrogenous bases: adenine A, thymine T, guanine G, and cytosine C.
And wait, they pair up in a specific way, don't they?
You got it. A always pairs with T, and G always pairs with C. And this base pairing is what creates the rungs of the DNA ladder.
Wow, it's like a code almost.
It is a code. The sequence of those bases determines the instructions for building proteins, which are what carry out all the functions of life.
Okay, so we've talked about carbon, biomolecules, DNA.
Mhm.
What else?
Well, let's switch gears a bit and talk about solutions and solubility.
Okay.
Ever notice how sugar dissolves faster in hot tea than in iced tea?
Yeah, actually.
Well, that's because solubility, or a substance's ability to dissolve in another substance, is often affected by temperature.
So like with the tea, the sugar's the solute and the tea is the solvent.
Exactly. The solute is what's being dissolved, and the solvent is what's doing the dissolving. And when they mix evenly, we get a solution.
Yeah.
And generally, the solubility of solids increases with temperature.
But I thought that wasn't always the case with gases, right?
You're right. You're thinking about how a can of soda fizzes when you open it. That's carbon dioxide gas escaping from the solution as the pressure decreases. For gases, solubility actually decreases as pressure decreases.
Okay, so it's the opposite. So what about concentration then?
Well, the concentration of a solution tells us how much solute is dissolved in a given amount of solvent. So a dilute solution has less solute, and a concentrated solution has more.
So like if you're making lemonade and you only add a little bit of mix, that would be dilute.
Exactly. But if you add a lot of mix, then it would be concentrated. And we can even calculate the concentration as a percentage. Like if you dissolve 10 grams of sugar in 100 milliliters of water, the concentration is 10%.
Okay, I think I'm getting that. What's next?
All right, let's add some soothing to our discussion and talk about acids, bases, and pH.
Okay.
You know that pucker up feeling you get when you taste lemon juice? That's the power of acids.
Acids, right.
Acids release hydrogen ions, or H plus L, when they're dissolved in water. And bases, they release hydroxide ions, OH, and we can actually detect these using indicators like litmus paper.
Oh yeah, I remember those from lab experiments. Acids turn blue, litmus paper red, right?
You got it. And bases turn red, litmus paper blue. And then there's the pH scale, which measures acidity and alkalinity. It ranges from 0, which is the most acidic, to 14, the most basic. 7 is neutral, like pure water.
Okay, so pH is super important for living things, right? Like acid rain can really damage forests and aquatic life because of its low pH.
Absolutely. And did you know that when acids and bases react, they actually neutralize each other? They form water and a salt?
Wow, I didn't realize that. So it's like a chemical handshake.
Yeah, I like that. Think of it like taking an antacid to soothe an upset stomach. That's a base neutralizing the excess acid. Pretty neat, huh?
Yeah, that is pretty neat. Okay, so I think we've covered a lot of ground in this first part of our deep dive.
We have. From the basics of carbon to those essential biomolecules, even a bit about solutions and acids and bases.
This is great.
And in the next part, we'll dive into some practice scenarios that might pop up on your test.
Perfect. I'm ready for it. All right, so let's dive into some practice scenarios that might show up on your test. We're not going to like memorize answers or anything, but we'll, make sure you really get how these concepts work.
Yeah, it's all about building that chemistry intuition.
Okay, so let's say there's a question about hydrocarbons. Something like, how does the structure of an alkene differ from an aromatic hydrocarbon? And how do those differences affect their properties?
All right, well, with alkenes, you've got hydrocarbons with at least one double bond between carbon atoms, so that makes them unsaturated. But aromatic hydrocarbons, they're cyclic, meaning their carbon atoms form a ring.
Right.
And they also have these alternating single and double bonds, which gives them this unique stability.
Oh, yeah, like benzene. I remember learning about that. It's got that strong smell, and it's used in things like mothballs.
Exactly. That's a classic example of an aromatic hydrocarbon. And that stability comes from the delocalized electrons in those alternating bonds. They kind of create this electron cloud that makes aromatic hydrocarbons less reactive than you might think.
Got it, got it. So, like, if you see a question about a hydrocarbon
Yep.
with a strong smell, or maybe it's unusually stable, think about those aromatic rings.
Right.
Okay, what about a question about cracking? How the petrochemical industry uses that process.
Okay, yeah. So cracking is all about breaking down large hydrocarbon molecules into smaller, more useful ones. It's kind of like taking apart a giant Lego structure and using the smaller pieces to build something new.
So instead of having one big, bulky alkane molecule, you can like crack it into smaller alkenes or even those aromatic hydrocarbons we were just talking about.
Mm.
And those can be used for all sorts of things like plastics, synthetic fabrics.
Right, or even fuels. Cracking lets us tailor those hydrocarbon molecules to what we need.
Okay, that makes sense. Now, switching gears to biomolecules. What if you get a question about why proteins sometimes have things like sulfur or phosphorus? Besides the usual carbon, hydrogen, oxygen, and nitrogen.
Hmm. Well, some amino acids, the building blocks of proteins, contain sulfur. And those sulfur atoms can form bonds that help stabilize the protein's structure, that intricate three-dimensional shape.
Right, right. And what about phosphorus?
Well, phosphorus is often part of phosphate groups. Those are super important for energy transfer within cells. Like with ATP, the molecule that provides energy for cellular processes, it's got those phosphate groups.
So even though sulfur and phosphorus might not be as abundant as like carbon, they still have these super important roles.
Absolutely. They're essential for protein structure and function.
Okay, now let's imagine a question about DNA replication, but like something goes wrong, maybe there's a mistake, and a thymine gets paired with a guanine instead of an adenine. What could happen then?
Whoa, that's a big problem. We know that DNA bases have very specific pairing rules, A with T and G with C. You can't just switch them around. It'd be like introducing a typo into a really important instruction manual.
Oh, yeah.
That kind of mistake could lead to a mutation, a change in the DNA sequence, and that it could change the protein that's supposed to be made from that DNA sequence. So you might end up with a malfunctioning protein or even a genetic disease.
Wow, so one tiny mistake can have huge consequences.
Exactly. It really shows you how important it is for DNA replication to be accurate.
Okay, let's switch gears again. Solutions and solubility. Imagine a question asking why oil and water don't mix, but ethanol and water do. Why is that?
Well, it all goes back to the idea that dissolves water is a polar molecule, meaning it has, a slightly positive end and a slightly negative end. And ethanol is polar too, so those molecules can interact and mix together easily. But oil's nonpolar. It
Yeah.
doesn't have those positive and negative ends. It's like trying to mix, I don't know, oil and water. They just don't want to hang out together.
They just don't mix.
Right. They'll stay separate and form those two distinct layers.
Got it. What about, what if you have a saturated solution of potassium nitrate and you cool it down from 60 degrees C to 20 degrees C?
Okay, well, we know that solubility usually decreases as temperature goes down,
Right,
especially for solids.
right.
So as that potassium nitrate solution cools down, it becomes supersaturated, meaning it's holding more dissolved solute than it normally could at that lower temperature.
So it's like too much stuff crammed in there.
Yeah, exactly. Like a party that's gotten way too crowded. Some of those guests got to leave.
Yeah.
So in this case, some of the potassium nitrate will probably crystallize out of the solution, form those solid crystals.
Oh, okay.
The solution is just trying to reach a new equilibrium, a balance between dissolved and undissolved solute at that new temperature.
Okay, now what about colligative properties? Like, how does adding salt to water affect its freezing point? And what's the reason behind that?
Well, we know that salt lowers the freezing point of water. I mean, that's why we sprinkle salt on icy roads, right, to melt the ice.
Right, right.
It's like the salt is getting in the way of the water molecules forming that nice, ordered structure of ice.
Ah, that makes sense.
Those dissolved salt particles interfere with the water molecules' ability to arrange themselves into that ice structure. So they have to get even colder before they can finally slow down enough to form ice.
Okay, I'm following you. Now, how about a question comparing hydrochloric acid, or HCl, and sodium hydroxide, NaOH, when they're dissolved in water?
Okay, so HCl is a strong acid. And when it's dissolved in water, it completely dissociates, releasing those hydrogen ions, H plus A, and chloride ions, Cl.
Okay.
And sodium hydroxide, that's a strong base, right? So it also completely dissociates, releasing sodium ions, NaPo plus, and hydroxide ions, OH.
So the main difference is that acids release H plus ions, and bases release OH ions.
You got it. And those ions are what give acids and bases their special properties.
Okay, cool. Let's try to bring this to, everyday life. What if there's a question about acidic and basic cleaning products, like how their chemical properties make them good for cleaning?
Okay, so a common acidic cleaner is vinegar. It has acetic acid. And it's great for cutting through grease and mineral deposits. The acid reacts with those substances and breaks them down.
Right, right. And for a
Mhm.
basic cleaner.
Well, there are ammonia-based cleaners. Those are really good at dissolving greasy grime.
Okay.
The hydroxide ions in those cleaners react with the fatty acids in grease, breaking them down into soap-like substances that can be rinsed away.
It's all about chemical reactions. Okay,
Mhm.
last one.
Yeah.
What about a question about buffer solutions? How they maintain a relatively constant pH even when you add a little bit of acid or base.
Ah, buffers. They're like the shock absorbers of the chemistry world. They resist changes in pH, which is super important for biological systems that are, sensitive to pH changes. Like our blood, it needs to maintain a pretty constant pH.
Right. So how do buffer solutions work?
Well, usually a buffer solution has a weak acid and its conjugate base.
Um, can you remind me what a conjugate base is?
Sure, it's what's left over from an acid after it's donated a proton or an H plus ion. So, for example, if we have acetic acid, its conjugate base is acetate.
Okay, I remember now. So in a buffer solution, if we add an acid, the conjugate base will react with those extra H plus ions, so the pH won't drop too much.
Exactly. And if we add a base, the weak acid will donate H plus ions to neutralize the added OH ions, so the pH won't rise too much.
So it's like this balancing act, keeping the pH stable.
You got it. It's all about maintaining that equilibrium.
Okay, now let's talk about some common misconceptions that might pop up on your test. Like, some people might think that carbon is only found in living things,
Yeah.
but we know that's not true, right?
Right. Carbon is in plenty of non-living things, like diamonds, graphite, fullerenes. Don't forget about carbon dioxide in the atmosphere either.
Oh, yeah, good point. And what about the idea that graphite is a carbon compound?
Nope, not true. It's a form of pure carbon, just like diamond, just arranged differently.
And speaking of pure carbon, another misconception is that all proteins contain sulfur.
Ah, yeah, not all proteins have sulfur. It depends on the amino acids they're made of. Some amino acids have sulfur and some don't.
Okay, now on to solutions. Some people think that stirring a solution will increase the solubility of the solute. But is that true?
Well, stirring doesn't actually change how much solute can dissolve at a given temperature. It just speeds up the dissolving process, helps to distribute those solute particles more evenly.
So it's like it's not making more dissolve, it's just making it dissolve faster.
Exactly. And another solubility misconception is that all substances become more soluble as temperature increases.
Oh, right. That's not always true for gases, is it?
Nope. The solubility of gases usually decreases as temperature increases. Think about that warm soda going flat faster than a cold one.
Yeah, that makes sense.
Okay, now for acids and bases. One misconception is that all ionic compounds form basic solutions when dissolved in water.
Oh, right. That's not always true either, is it?
Not at all.
Yeah.
It all depends on the ions that are released when that compound dissolves. If it releases H plus ions, the solution will be acidic. If it releases OH ions, it'll be basic. And if it releases neither, it'll be neutral.
So it all depends on those ions.
Precisely.
And finally, there's the misconception that the strength of an acid or base is related to its concentration.
That's a tricky one. The strength of an acid or base actually depends on how much it dissociates in solution. A strong acid or base will completely dissociate into ions, while a weak acid or base will only partially dissociate.
So concentration tells you how much acid or base is there, but strength tells you how much of it breaks apart into those ions.
Exactly. Two different things.
Wow, we've covered a lot of ground in this second part. We've revisited concepts, talked about practice scenarios, and even busted some misconceptions.
It's all about making sure you really understand these concepts.
Absolutely. And in our final part, we'll wrap up with some test-taking strategies that can help you ace that chemistry exam. All right, so we've gone through a ton of material, revisited those tricky concepts, busted some misconceptions. I think we're ready to talk about test-taking strategies, how to actually ace that chemistry exam.
Yeah, let's give you some practical tips and tricks to help you tackle those multiple choice questions and those problem-solving challenges.
Okay, so first things first, read each question carefully, right?
Oh yeah, absolutely. Sometimes one word can change the whole meaning of a question. Don't rush. Make sure you really get what's being asked before you jump into answering.
And I know it sounds basic, but it's so easy to miss those little details when you're, under pressure.
Exactly. It also helps to underline or highlight those key words or phrases in the question. So you can really focus on the important stuff and not get lost in the extra details.
That's a good tip. And another one is to try and eliminate those obviously wrong answer choices.
Oh yeah, that's a classic. Even if you're not 100% sure of the right answer, you can often narrow it down, right?
Right. And if you get totally stuck on a question,
Don't dwell on it forever. Move on and come back to it later. Sometimes just taking a break and coming back with fresh eyes, it can, help you see things differently.
that's great advice. And of course, don't forget to review your notes and practice problems.
Yeah, it's like a warm-up for your brain, right? Get those neural pathways firing.
Exactly. Okay, so let's talk about some specific types of questions. You're probably going to see a lot of multiple choice questions, testing definitions, concepts, relationships, all that good stuff.
Yeah. And with those, it's super important to pay attention to those little differences in wording. Sometimes the answer choices will seem really similar, but only one is actually correct based on, how the question is phrased.
Right, right. And then there are those problem-solving questions where you have to actually apply those concepts and formulas.
Yeah. Take your time with those. Show your work step by step and double check those calculations. It often helps to draw diagrams or write out the steps just to make sure you're not making, any silly mistakes.
And don't forget about units.
Oh, yeah, units are crucial. Make sure you're using the right ones throughout your calculations and make sure your final answer has the correct units too. A missing unit can mess everything up.
Absolutely. Okay, so let's shift gears a bit and talk about some general test-taking tips. Those things that can help you stay calm and focused.
Yeah, like getting a good night's sleep before the test. I mean, your brain needs rest, right?
Oh, yeah. Cramming all night is definitely not the way to go.
Nope. It's better to, review the material over time consistently and eat a healthy breakfast on test day.
Yeah, your brain needs fuel, just like your body.
Exactly. And during the test, just try to stay relaxed. Remember, you've prepared for this. You've got the knowledge. You can do this. If you start to feel anxious, just take a few deep breaths and refocus.
And most importantly, believe in yourself. You've put in the work, you know the material. Trust your instincts.
And remember, even if there are a few tough questions, don't let them get you down. Just keep going. Focus on the ones you know.
Every question you answer correctly is a step closer to acing that exam.
You got this.
We've covered so much in this deep dive. I mean, from the basic building blocks of matter to those incredibly complex processes that make life possible.
And we've seen how chemistry, connects to the world in so many ways.
It's pretty amazing when you think about it.
It really is.
So as you head into that test, stay curious, keep asking questions, and don't be afraid of the challenge.
The world of chemistry is full of incredible discoveries. Who knows, maybe this deep dive has sparked a passion in you that'll lead to a career in science.
Well, that's it for our deep dive into chapters 9 and 10. We hope this was helpful and that you feel confident and prepared to rock that chemistry exam. Good luck, and remember, the world of chemistry is waiting to be explored.